Let's get to the marks. We're getting involved with developing drugs and the main stages in drug testing. So, pharmaceutical companies start their drug testing in the preclinical trials or testing. This means before the clinic, so it's not tested on humans. They're testing the drugs or medicines on animal cells and tissues. And that means using a microscope and examining the effect of the medicine on the tissues. Does it make the cells burst? Does it damage the cell membrane? Does it make the cytoplasm leak out? Does it affect the DNA inside the nucleus? These are all things they will be looking for. If it passes this phase of the development, they will move the testing on to testing on animals, which will then see how does it affect a whole organism. So if it hasn't damaged the cells, will it damage the whole organism with all those organ systems like the circulatory system and respiratory system? Will it affect any of those in an animal? And that's the second stage. They're really looking for efficacy, which means does it work, toxicity, how harmful it is, and what dosage they should apply. Now, if it survives this stage, and by the way, some people think it's cruel to test on animals, and that's a legitimate concern. But in the United Kingdom, the law states a drug must be tested on two different species of live animals. Take these cute little bunnies here or these cute little kittens. The drugs could be tested on them, and they'll look to see not only do the animals survive, but have there been any negative effects on any of their body systems, their blood pressure, their breathing, anything like that. If it fails at this stage, the drug will be ditched. Now, there are people that protest and they believe that it's cruel to test drugs on animals and you shouldn't do that. Some people would argue it's better to test on animals than human volunteers when the drug could still be dangerous. So it's better that an animal dies than a human. Anyway, once it passes the preclinical trials, we move into the clinical trials. These will take place in a hospital with doctors to monitor the patients or the volunteers. And the first stage is the testing on healthy volunteers. Why healthy? Well, if you test the drug and it has bad side effects and you test it on people with a weak heart or who are ill and their immune system's low, it's going to be a lot worse. So first you test on healthy people who volunteered. You use a low dose, and if there's no side effects, we can move to the clinical trials, step 2. Now we're going to test on sick people, people whose immune system may be weakened, and we're going to see what the optimum dose is. What's the best amount that gives the least side effects for the greatest impact on the disease? Let's say we're testing a new drug to get rid of migraines or headaches. We're looking to get rid of that headache with the least side effects, the least raising in blood pressure, nausea, or whatever those side effects may be. And that's the stage, that's the clinical trial step 2. Now, it's also important to bear in mind that we often carry out blind trials. And what that means is the patient won't know if they're being given a real drug or a placebo. There's something called the placebo effect. I get this myself. I take a paracetamol when I have a headache, and sometimes within 30 seconds, I think, oh, the headache's feeling better. It's going away. But actually, the paracetamol is impossible for it to got into my system, dissolved, and be put into my blood by that time or had an effect. This is known as the placebo effect, the psychological effects of taking drugs. So we want to account for that. So we give half our patients the placebo pill, which has no drug in it. You'll be asked, what is a placebo? It's a pill with no drug. And we give the other half the actual drug. And what we're looking for there is to see if the effects are real of the drug. And now a double blind trial is much more common than just a blind trial. A double blind is that the doctor doesn't know and the patient don't know who's on the correct pill, who's on the medicine and who's on the sugar and wheat or the flour pill. So the idea here is that the doctor could accidentally cause bias in the trial, spelled BIAS, bias. If the doctor knows, they could accidentally bias the results. Somebody has to know, and that's the scientist behind the trial. So the scientists behind the testing will know. They will send the doctor the red and green pills. Half of them are placebo, half of them are the real drug, and they'll also hand them out. And the scientists will analyze the results. All right, guys, let's get into some exam questions. So pause the video Have a go at these questions. Right, I'm going to go through the answers. Well, I'm not going to go through them. We're just going to put the answers up. And what you should do is check your answers. I recommend at the end of my videos, there are exam questions. I recommend grabbing some scrap paper, grabbing a pencil or a pen, and scribbling down your answers, and then you can check them off. So pause the video. Okay, here are the answers. So check them, see how you did. These are all exam questions, real exam questions that people had in their GCSE biology. So we've got a couple more questions coming up. So pause the video. Okay, now it's time to check your answers. See how you did. I would always watch my videos with a scrap of paper and a pencil. Here we go. Pause. And finally, check your answers. And don't forget, guys, subscribe, like. I'm making videos to help you smash those exams. So don't forget, let's get to the marks.